The central dogma describes mRNA synthesis using a DNA template, known as transcription, a precursor step to protein synthesis by translation. RNA polymerases are multi-subunit complexes responsible for RNA synthesis, which synthesize the RNA transcript in the 5' to 3' direction. Eukaryotes have 3 different types of RNA polymerase, with RNA polymerase II transcribing all protein-coding genes. The TATA box has a consensus sequence consisting solely of bases adenine and thymine, found upstream of the transcription start point. The TATA box has the responsibility of indicating which DNA strand will be transcribed, fixing the site of initiation, and binding RNA polymerase through the use of general transcription factors. Transcription factors are proteins which control the rate of transcription. Examples include activators, repressors, and general transcription factors. General transcription factors are sequentially assembled for a pre-initiation complex necessary for transcription initiation. Transcription starts with the binding of TBP to the DNA, inducing bending of the DNA chain, which polymerase preferentially binds to. The bend brings DNA into contact with TF2B, which positions the promoter DNA correctly and guides DNA through the complex. RNA polymerase II then binds to the complex before the complex moves along the DNA and reaches the transcription start site. TF2H is then recruited to the PIC, which has helicase activity and unwinds the DNA. The template strand then moves through the PIC, and the first base to be transcribed enters the polymerase active site. Initiation can now begin, in which an RNA nucleoside triphosphate complementary binds to the DNA template strand, followed by elongation and synthesis of the RNA. Elongation occurs via production of phosphodiester bonds, synthesizing an RNA nucleotide polymer using nucleoside triphosphates. Once the RNA strand is around 20 to 100 nucleotides long, RNA polymerase II can disconnect from other components of transcription machinery, giving rise to a fully functioning elongation complex.
A trigger loop located near the polymerase active site repositions when correct base pairing occurs, causing RNA strands to be specifically complementary to the DNA template and causing elongation by inducing formation of phosphodiester bonds. As each bond is formed, polymerase moves forward, bringing the next template nucleotide into the active site. The RNA-DNA hybrids are then separated and the 2 DNA strands rejoined. Elongation continues until the polymerase reaches a termination sequence. The newly synthesized RNA strand, RNA polymerase II, and DNA strands all separate. There are multiple mechanisms of transcription termination. Intrinsic termination occurs when the RNA folds to form a hairpin loop at the termination sequence. The hairpin loop disrupts the RNA-DNA hybrid and prevents further elongation. The next involves a protein factor. In bacteria, this is called a raw-dependent termination, as it requires a raw protein which inserts into the main channel of the polymerase complex and separates the RNA-DNA hybrid. Repeated transcription can occur once RNA polymerase has polymerized 50 to 60 nucleotides. Post-transcriptional modifications occur only in eukaryotes. These are capping, polyadenylation, and splicing. Pre-mRNA is formed first in the nucleus, which contains both introns and exons. The 5' end of mRNA is altered by the addition of 7-methylguanosine. This terminal group is called a cap, necessary for translation. Next, the 3' terminus is first modified by exonuclease digestion before the addition of polyadenylate sequences. This is known as a poly-A-tail and helps maintain mRNA stability. Once capping and polyadenylation has occurred, alternative splicing can occur, in which introns are removed and exons are rejoined in alternative sequences. These are called splice variants, allowing for increased complexity without increasing the number of genes of an organism, which provides better utilization of genetic material in eukaryotes compared with prokaryotes.